to the cloud. Okay, excellent. All right, so everyone can see my screen? You guys here? Everyone remotely can?
Yep.
Excellent. So I would just like to start off, we got like another minute or two, but while we're waiting for people to show up, I would just like to make an announcement here. Remote Matt Damiano, who's at the CMAS, who's one of the remote students for this class, he just received this fellowship from Sea Grant. And so this news article came out yesterday or the day before that he's one of the newest fellows selected for National Marine Fisheries Service and National Sea Grant College Program for his project. So I just like to give you a shout out there. Matt's working on population dynamics here. Looks like Kobe and things like that. So if you look this up, you can read more about it, and I'm sure Matt would love to let you know more about that. So just wanted to point that out. And with that, I'm going to minimize this. Because we didn't have time last time to go over the syllabus, I'd like to go over that real quick.
Recording has begun.
Very good. So the course syllabus here, this is some information about these recordings that Delta is going to do. I was told that they're automatically recording these, and so this is the link that you can get those lectures at. If you have any questions, you can email here or look up information here at this site, right here. This is the course description from the catalog and also learning outcomes. The textbook is this one right here, Diversity of Fishes. And it's a pretty reasonable book. Some of the readings for the class, really the lectures are taken from this book. So throughout the syllabus, there's the readings that are listed there as well. Grading policy is simple. There's 2 exams that are graded. There's a scientific research report, it's worth 100 points. And then you all have some kind of classroom participation here, 100 points. And then there is a cumulative final exam. Now that's scary, right? It's cumulative. So this is what I do. There are no makeup exams in this class. You will actually be given 3 exams. I'm only going to grade the best 2. And then they will be returned to you, and you will use them as study tools for your cumulative final exam. Does that make sense? So the cumulative final is tested on everything. It's not going to be questions identical from the other 3 exams, but they're going to be very much the same topic. So there is no makeup exam, because if you can't make an exam, we just give you a 0 and give you the blank exam, and you can use it to study for the cumulative. Does that make sense? Here's the course breakdown. Adaptations. We'll still have the exams on one day here. If you're a remote person, like at CMAS, we'll send you the exam by email, and it will be proctored at your institution. But Linnea, your TA, is going to proctor half of you students in one room, and I will proctor the other half in this room. So on exam days, everybody shows up. Does that make sense? Classroom participation is what it is. Feel free to speak up. We like to talk about fish here, and so if you find an interesting article in the news about fish, please let me know, bring it up. And if I ask a question, please chime in. This is sort of the rubric breakdown there. There is a scientific research report. And really, I'd like to get these topics submitted no later than exam 1. Now, what that means is I'm not even going to beat around the bush. One of the questions on exam 1 is going to be an essay. What is your research paper topic? So it will be on the exam. And so explain to me a little bit about what that'll be. The topics are due at the end of the semester. And what I'm looking for here is a 5 to 8 page long paper. Really, you can have a few peer-reviewed references in there. We don't really want like a 20-page long paper. What we really want is just tell me something interesting about fish that I don't already know. That's your challenge. And so throughout this course, you're going to have these little stories that I tell, and those are oftentimes based on previous students' papers. And so at the end of every lecture, there'll be a little story that I'll go into, and oftentimes those were previous student topics. No extra credit in the course, please do not ask. This is all then just regular stuff from the university, attendance policy, late assignments, academic integrity, et cetera, et cetera. And so please just do your best to attend. This semester is a little bit crazy because of the virus, so I'm going to be pretty lax on this attendance policy. That's why, you know, if you show up, you can, and also that's why we got the remote option as well. So that's that. And then I always have to mention this. Please complete your online course evaluations at the end of the semester when you get prompted. And also there's the Thank a Teacher program if you want to look into that as well. And so with that, here we are into the syllabus. So first day of classes was 2 days ago. Today we're going to be talking about diversity of fishes and systematics. I'm going to go over these ancient fishes here. And these were the readings from the book that correspond to today's lecture. So chapter 11 and 13. Sometimes I might go over some of these readings if I have time. I'll be glad to answer questions about them. But this remote option sort of makes it difficult for me to bust out my textbook and show the remote people what's actually in the reading, because I don't have an electronic copy of the book. So with that, exam 1 is scheduled here, September 8th. Exam 2 is here, October 13. And what it is exam 1 is going to cover lectures 1 through 8. Exam 2 is lectures 9 through 17. And exam 3 here in November 12th will be lectures 18 through 24. And also here is when your reports are due. And so the information here for the final exam, I'm going to split you guys up into 2 groups, just like you are with the lectures. The final exam will be held between 3:30 and 6 p.m. in this room. And so the Group A students are going to show up from 3:30 to 4:45. Group B students are going to show up from 4:45 to 6. So I'm going to give 2 final exams, one after the other. And that's because it's a group final exam that is cumulative. Does that make sense? And I'll give you more information on that. Also, the final grades are due by me to the university on November 23rd, which means I cannot take any assignments that are late after that. My grade has to be submitted, otherwise I get in trouble. So late assignments, that's the 0 hour, so to speak. So any questions there on the syllabus? Pretty straightforward, right? So with that, I'm going to get rid of this, and we will start with the lecture. So everyone has these in PowerPoint, so you can feel free to access these at any time. I'm just going to go through here real quick to show you here. I have video links embedded in here. And so you can rewatch these videos. You just click on it, it takes you right to YouTube, and you can watch the videos. So you've got this, I'll show them today, but if you want to go back, you've got these notes. You can access all these videos and you can look at them. And so this is the second lecture here. End of last lecture, I gave you this this tree. I said this is going to be the outline for the taxonomy portion of the course. This is going to be pretty much the course 1. There'll be a few questions on exam 2 once we get down here to the bony fishes, the teleosts. But we're basically going to start up here with the less derived fishes, and we're going to move down. And so today, we're going to be talking about all these things. These things here, if we follow this line down, the placoderms will be the last ones we talk about. If we follow this all the way down to Gnathostomata, these fish, none of them have jaws, right? Most of these things are extinct, actually. Any of these ones with the little cross on there are extinct. The lampreys are extant, meaning they still live, and the Myxiniformes are extant, the hagfishes. Other than that, everything else here is extinct. But we have to understand these things because they have features about them that help us to explain more derived fishes. So we're going to start here, and so at the beginning of every lecture, I'm going to remind everyone where we are in this tree. All right? Any questions there? So we're looking at vertebrates, which means they have a backbone, but generally speaking, no jaws until we get down here, the gnathostomata. Gnathostomata means to have a jawed mouth. So this is the origin of fishes. And anyone who's taken a biology course, right? Have you gone like intro to biology? Well, when we look at these earliest animals, and I'm going to go ahead and, where is it at? Here. Can you see that better? These things here, what is this? It's a bryozoan. These are ancient animals. Now, these are benthic. What does the word sessile mean? Anyone know?
They don't
Exactly.
move.
What was that online?
Probably the same thing from not online, since you said exactly.
Remote math. Very good. To not move. I know a lot of students who are sessile. I'm just kidding. There's a lot of faculty who are sessile. These things, you know, the first invertebrates, they had these gill-like projections here, right? And, you know, these things gave rise to primitive echinoderms, like sponges and things like that, and also then these pterobranchs, which also were sessile. And notice these external gills. Number one, they're external, and number two, these were primarily for feeding, right? And you're going to find out later on that the gills in the fish are still involved in feeding. So there was these external gills, then there was a shift from arm-feeding gill filter feeders to the gills then being enclosed on the inside of the organism. So that instead of them being outside, they're then enclosed. So we have things like acorn worms and then tunicates, these sea squirts. These things had a sessile adult stage with a free swimming larval stage. So these things are all sessile, external gills, primarily for feeding. Now we see enclosed gills with a free swimming larval stage and a sessile adult stage. Then we go into what we call the advanced chordates, where instead of the adults being sessile, they also are mobile. So it's not just a larval mobility stage, there's also mobile adults. And again, the gills are enclosed. So if we look here, here is one of the one of the least derived chordates, the lancet, or the amphioxus. Now, look at this thing. It's not a fish, but it looks pretty close to it. You do see some of these little gill filter feeding apparatuses here. This is called the wheel organ. It's a feeding apparatus of these gills right here, but there's also these internal gills here. And so this is where we start seeing maybe a differentiation of maybe feeding is going on here and gas exchange is going on here. And so these primitive filter feeding vertebrates gave then rise to ostracoderms, which is the ancestral fish. Ostracoderm means tough or plate-like skin. And so here is one of those early fishes, an ostracoderm reconstructed from the fossil record. Look at this. Looks very similar to amphioxus. The difference is this thing is transparent. You can see through it. It's a very fleshy, thin skin. This thing is armored with bone. It has these very thick plates on it that armored it. So this is where we start seeing the beginning of the fishes, heavily armored creatures with internal gills that were originally designed for feeding, but also now are being used for gas exchange. All these ostracoderms are extinct. So all we have are the fossils to talk about. These guys are still living. You can go down to Florida and find these, but these guys are extinct. All right, so that's sort of the beginning of the fishes. So here's another phylogenetic tree, but it's sort of laid out this way. And so those ostracoderms I'm talking about are right here. Here they are. And a lot of times the armor sometimes was over just the head, and the tail of it didn't have skin or scales. Every now and then, like here's another head shield one. Sometimes they were fully armored all the way down. And you see that these things sort of arose during the Ordovician or the Silurian 450 to 500 million years ago, long time ago. But they were all extinct here. This is what the end is. The start of the line is they become apparent. Here's when they leave the fossil record. The Devonian. Now, if I was a student studying for this class, I might expect a question on an exam such as, what is the age of fishes? And that would be the Devonian. Because even though these things started out in the Ordovician and the Silurian, a lot of them went extinct, but we see a major radiation here in all these other fishes occurring during the Devonian. And then we see really another second one that occurs down here maybe later during the Triassic. But generally speaking, there's a major radiation of fishes here. And even before these things went extinct, these things were coming about. And I'm going to tell you the reason. A lot of these things, look at this trend here. No jaws versus jaws. The jawed stuff got rid of the no-jawed stuff, right? Whether they ate them or outcompeted them, that was a major advancement. And so here we see elasmobranchs like sharks, rays, and skates starting to show up. There's a dashed line here because, you know, if you look at your reading, chondrichthyan scales and denticles are found in the late Ordovician deposits, but these are cartilaginous fishes, so they don't have very good bones that are preserved. So it's kind of hard to tell really what was going on there, hence the dashed line. So a lot of times what we say is the acanthodian fishes were the very first. And these things then were extinct sometime in the Permian. The placoderms are also plate-scaled fishes. Again, this heavily armored head with kind of a naked body. The dipnoi, still in existence. The lungfishes, look at these things. 400 million years ago, they started off, and we still got 6 species around. The coelacanths, 2 species. There's actually 3 now. They found another one. Accipenteriformes, the sturgeons, 27 species coming about during the Triassic. Polypteriformes are the African reed fishes, 12 species. Neopterygia, I don't really like this term. It sort of is a non-used term, and really we'll skip down here to the gars. 7 species, all freshwater, only in North America. Ameiformes, the bowfin, 1 species, only in North America, freshwater. And then look at this, teleosts, 26,000 species. So again, if you're going to guess, oh, look, we got 26,000 of these. So if you were to say, if I gave you a random picture of a fish and said, what group is it? I mean, teleost is a pretty good guess. Holocephali are the rat tails. There's only a few of those. And then the elasmobranchs, there's about 600 species of elasmobranchs. So sharks, rays, and skates. And we go up here, the only remaining fishes with no jaws, about 36 species of hagfish, 42 species of lampreys. So Petromyzontoideae and Myxonoidea. That's all we got left. And so these are your eras, these are your periods, these are some of the taxa and where they showed up. So we're going to dive right in here with these guys. But again, a major trend here. When jaws came around, the guys without jaws did not hang around for very much longer. So, any questions there? Here is Amphioxus, not a true fish. It is a true chordate. It has a notochord. This little orange line down its body is a notochord that helps protect the nervous system. It is a prevertebrate, right? So these things don't have true bones, but they do have a notochord. They're very small, about 1 inch long. And every now and then they'll have these in intro to biology classes. You can look at them under the microscope or you can dissect them. There's a nerve cord that runs along the notochord. Filter feeders. Here again is this wheel organ that allows the thing to feed with these external cirri, these buccal cirri. This wheel organ allows it to feed, and then there is some flap here, the velum, that allows that filters water to capture and remove unwanted particles. So there's a mucus that's on here, and that way the organism can sort of decide what it wants to eat. It doesn't have to filter everything. And then there's this gill basket that also helps feeding up here. And these things sort of live in the sand, but you can see they're sort of transparent, kind of fish-like, but no bones. So they're not really a fish. They're Amphioxus is the genus, and their common name are the lancets. So those are like sort of the earliest of the early things. The ostracoderms, shell-skinned or thick-skinned. There's a bony shield. Look at these things. They look like UFOs, man. These bony heads, and then they've got these tails that are almost naked. There's no scales on them. Jawless, no jaws, so they're agnatha. And they're really not a valid taxonomic designation. The previous slide I showed, there's several things that I just sort of collectively called ostracoderms. And so that's what we just call these ancient fishes together. They're today represented by superclass agnatha, the hagfishes, the pterospidomorphs, and then the lampreys, cephalospidomorphi. Really, pterospidomorphi is not really used all that much. I like to call them the mixini. These things have lost a bony shield. And they're also more highly specialized. So hagfish and lampreys do not have this body design. These ostracoderms were the first true vertebrates. This was bone. These bony shields were exactly what they are, bone, right? So they are vertebrates by definition. That sort of defines a vertebrate. They were dominant in the Devonian, again, the age of fishes, 440 to 350 million years ago, most extinct then by 250 million years ago. These things did not have well-defined gill arches. They actually had gills and muscular pouches along the sides of them. So like along the side of these things, you see these little spots. Those were like little holes in the bony shield that led to a gill pouch. These were primarily benthic filter feeders that scavenged on stuff. So they had no jaws. They just sort of hung around in the muck and ate basically whatever was on the bottom of the ocean. Very short, usually about 30 centimeters, no longer than that. And there were, again, two living classes representing them, hagfishes and lampreys. So these things are pretty crazy. Order Myxiniformes. Look at this thing right here. This is a hagfish. Can you see? I'm sorry. You can scoot if you need. This hagfish, look at, again, this isn't a jaw. This is what it looks like with its mouth closed. And then it opens its mouth and bam, look at these teeth. Crazy. Single family, Myxinidae. So when we're studying for the exam, I'll ask you a multiple choice question on families of fishes. And I'll ask you characteristics like the ones that are listed here. And then I'll give you 4 options. Which family is it? And so you'll have to know, oh, it's Myxinidae. And it'll be something like, what has 4 hearts, 11 pairs of gill exits, a cloaca, an everted tongue, and produces copious amounts of mucus? What family of fishes does that? And then you would answer Myxinidae. And you've got a multiple choice example there to pick from. So when you're studying these slides, these are the characteristics to focus on, and this is the term to remember. So 5 genera of hagfishes, 36 species. These are all marine, no freshwater. Eel-shaped body. Look at this thing, eel-shaped. That's exactly what it's called, eel-shaped. Even though they're hagfish, it's unfortunate. The hagfish came first before the eel, but we always give props to the eel and not the hagfish. You should have said hagfish-shaped body, but that's not how we do it. These things are scavengers. Some are predators. Like these teeth actually can be used to do some damage. Anterior tentacles, no dorsal fin. So there's no dorsal fin on these things. They've got these tentacles here. They've got 4 pairs of them, and they're sensory. So these things can smell and taste with these tentacles. There's a single nostril here at the end that's used for breathing. It's inhaling water here. The mouth is really just for the intake of food. They have 4 hearts, 11 gill slits along the side. They have a cloaca, and then these teeth, these things actually, you know, you've heard of these whale falls where the whale's carcass dies and sinks to the bottom. What happens is these hagfish then get in there and they use their teeth to burrow inside of the whale, and they just sit there and feast on the rotting carcass of the whale. That's what they do. So they can enter the abdominal cavity and eat a fish from the inside out. But they don't do this to living fish. It's always dead ones. But they can take a bite out of things. Typically, these things mostly eat underwater invertebrates, polychaete worms and things like that. So this is family Myxinidae, the hagfishes. So Hagfish respiration. Water enters this nostril here in the front of the head, and then it comes in, and then there's a little flap of skin here called a velum, and the water is diverted then into these gill sacs. And you can sort of see these little spots along the side. These are these gill sacs. So water comes in through the gill, and then out through the side of the fish. So it comes in the nose, into the gill sac, and then out the side of the fish, right? And they could have 11 of these things along the side. The mouth is only for taking in water. And so this little flap here closes so that food doesn't accidentally go into the gill chamber. But if the hagfish wants to, it can put water in and then blow it out its mouth if it needs to do that. They can also respire through their skin. And so the mouth is not involved in respiration. Diet is oftentimes worms. They have very slow metabolism. These things live on the bottom of the ocean where it's really cold, and they can go for months without eating. They just sort of tumble around on the bottom of the ocean, eating dead stuff. That's their life. That's the life of the earliest fishes. And if you look at like the placoderms and these ostracoderms and things like that, they did pretty much the same thing, tumbling around on the bottom. Hagfish reproduction, they have direct development, which means the young hatch as miniature adults, fully formed. So this is a hagfish egg. Look at this thing. These are little hooks. So when the hagfish eggs are laid, these little hooks allow them to sort of form chains or attach to aquatic vegetation or substrate so they don't get carried away by currents. And inside here is a miniature hagfish. When they hatch, they hatch fully formed, which is what direct development is. There's no larval stage. That's odd because almost all other fishes have a larval stage. They're hermaphrodites. They have both ovaries and testes that are present in the same individual. So we're talking simultaneous hermaphroditism. But they only turn one gonad on at a time. They don't self-reproduce with themselves. So they sometimes will act like a female, sometimes act like a male. They can sort of switch it off. These eggs are about 1 inch in length. They're very leathery. And again, they have these little tips there that attach. And when they hatch, this thing cracks right along this little line. That little lid comes off and the baby hagfish swims out. Pretty cool. And so those are hagfish. Look at these things. Their common name is slime eels. This is the amount of slime one of these things can make. Look at that. This guy is holding this hagfish, and look at the slime. So along towards the head, they have these gill pores, 11 of them. And then these little dots that are down the side of them here, these are mucus pores. So some of these, the first 11 are used for gas exchange, and the other ones are used to produce this mucus. And they can do this like that. They can do it in a matter of seconds. They also form these knots, and they're very slimy. So if you try to grab a hold of one of these things, what it does is it'll form a knot at its tail, and then it'll work that knot up towards the head, and it will wiggle. It'll use that knot as leverage to pull itself free from your hand. You can't hold on to it. So it's a defense mechanism. So I've got a video here that I'll show you of the hagfish. And so this is the life of a hagfish. Now, we tested the sound on this earlier, so please, all the remote people, let me know.
Oh, there's a hagfish.
Look at these things.
You see that?
Oh,
its
yes.
eyes shining
Let's
out
zoom
of
in on the evil hagfish.
evil creature.
Full zoom. Oh, I don't know if I want to go in that far.
Tying into a knot.
Great shot.
Yeah, the skin on it is really kind of
So gross.
Oh wow, so it's not eyes. It's
We
kind of
brought them up
just...
in bait traps, and they were kind of like crawling out of it
Yeah.
on the front porch of Herc.
We tried to get them with bait traps a few years ago with ONC, and they, I don't know if they changed the bait trap, but they kept getting out at the time.
Yeah, they've been working on those bait traps. They're trying to get crabs, and they haven't been successful, really.
We're getting some glimpses of its mouth.
Yeah, how great. And I think you can...
Yeah, wonderful. This is excellent.
Yeah, I remember someone saying, oh, they don't have eyes, and every time we've passed them, I've thought that those spots were its eyes, but it's eyes. Obviously, it's not. It just appears to be.
Yeah, they have no
Go.
eyes.
Looks like it's going
Look at
for
that.
the
It's
knot.
nodding itself up. Look at that. Just
Oh,
tumbles
wow.
away.
Wow.
Oh no! Wow!
Oh, that's so great.
What
They
a
call it a
terrible
mouth.
thing.
It's really simple.
Okay, guys, what's the plan?
All right, what do you guys want to do?
That's it. That's all these
I wanna look
things
at these
do throughout
hagfish.
their life. Tumble around on the
That's
bottom
Yeah.
through the muck.
Can I zoom out now?
All right, full wide.
Look at them all out there. Now, you want to see them make slime? Check this out. So this is an underwater camera that they put a bait bag on, and they show sharks coming up trying to eat these things. Watch how fast these things make mucus. This video here has no sound because it's, I don't know, thousands of feet underwater. So there is the bait. So look at that hagfish. Watch this shark. Look at the shark's mouth. Look at the mucus. Shark did not like that. Here's another one. Yeah. He tried to get that hagfish, but nope. Look at the mucus that shark is dragging behind it. That's how much mucus they can make in a very short period of time. Totally a defense mechanism. None of them ate a hagfish. That's it. And so those are the hagfish slime defense mechanisms. And so if you take a hagfish like this and put it in a 5-gallon bucket and stir it around, the entire bucket will be filled with slime like that. So defense. And you can see that the predators do not like that. So that is a defense mechanism for the hagfish. So very interesting creatures. You see them tumbling around in the bottom of the ocean, producing slime. That's their life. All right. Any questions there? Family Myxinidae. Here's another one, single family, Petromyzontidae, the lampreys. 10 genera, 42 species. Adults typically do not feed. What? Everyone's heard the story of the lampreys, the parasitic lampreys. Most lampreys actually do not feed. Only the sea lamprey does. Most of them, actually, it's an adult stage that they metamorphose into, and they go on a spawning run, they reproduce and die. So most lampreys actually do not feed as adults. The sea lamprey is an exception. These things reproduce after metamorphosis and then die, typically. They do have functional eyes. So the hagfish has no eyes, these things do. Look at, this is a very clear eye here. Very clear eye here. Their mouth actually is typically used to hold on to rocks. Sea lamprey is designed to hold on to other fish and eat and parasitize them, but most of the time lampreys use this oral disc to hold on to rocks. Because a lot of times these things live in freshwater rivers. And so they attach to rocks to avoid being pulled downstream by the current. There is a dorsal nostril. Unlike the hagfish, this nostril is not involved in respiration. It's involved in, or it is involved in respiration, sorry, but also in the sense of smell. They have 7 pairs of gill slits along the side. Now, these things are blind pouches. Water goes in and comes out. So they don't use the head at all for respiration. The mouth is used to attach to things. The nostril is used for the sense of smell. And the gill pouches themselves have muscular pumping apparatus that allows water to go in and out. These things have a larval stage. So unlike the hagfish, which was direct development, they have this ammocoete. Whoa, look at this thing. Looks just like Amphioxus, right? This is what you call that phylogeny, or what is it? Ontogeny recapitulates phylogeny thing. So these things look just like Amphioxus. And so lamprey live like this for several years. Then they metamorphose into the adults, reproduce, and typically die. The sea lampreys will actually act as a parasite for a few years before they reproduce and then die. But typically speaking, that's their life cycle. So if we look at freshwater lampreys, most are not parasitic. Freshwater species are not parasitic. The American brook lamprey, for instance, here is not. It is 4 to 8 inches long. There's a dark blotch here on the tip of the caudal fin, and it's usually deeply notched right here. That's sort of an indication that this is a freshwater lamprey, in this case, an American brook lamprey. And that's in a comparison to the famous Petromyzon marinus, the sea lamprey. Look at this. This thing here is the parasitizing disc, right? This thing is a bunch of horny teeth. It's designed to attach to the side of a fish, and then there's this rasping tongue here that can scrape the side of the fish. And once the hole is made, then the parasite drinks the body fluids or the ground up tissue. It eats it. These are large, up to 47 inches long. They are parasitic. They are anadromous. They live in the ocean, come into rivers to spawn in the fresh water. The dorsal fin here is clearly separated into 2, what we call an anterior, close to the head, posterior, farther away, close to the tail. And so these things are parasites. And there's 1, 2, 3, 4, 5, 6, 7. There are your 7 gill slits. And again, water comes in, goes into the gill, and then gets pumped out. And notice, look at this. It can actually close that oral disc. It can fold it in half. So here it is fully open, using it to attach to something, and here's a lamprey folding that thing shut. So they can actually control that. And so here are these ammocoetes. Look how small they are. And then here are some sea lampreys attaching themselves to these rocks in a river. And so one sea lamprey female can produce 250,000 eggs. Spawning migrations occur from March to June from the ocean into freshwater rivers, and they can make mud nests or nest in gravel rock substrate. And they pair spawn, so a male and a female will spawn together. So unlike the hagfish, which is a hermaphrodite, that they also pair spawn, but the sea lampreys are actually what we call dioecious. There's one gender that's dedicated. Eggs hatch into the ammocoetes, which behave much like Amphioxus. These things just swim around filter feeding. And then at some point, after 3 to 7 years, they will metamorphose into the adult form. And adults can actually live up to 2 years as a parasite then before spawning, and then they usually die after spawning. I think every now and then a lamprey might live, but typically they don't. These spawning migrations are pretty tough. There's not very good survival. And so here is a video showing lampreys using that oral disc to attach substrates. You can see these things swimming through this little river, creek really, not very deep. You see they're using that oral disc to attach to the stone. Sometimes they get a little feisty like that. That rock isn't big enough. Like that. If they use it as a handhold to hold on to avoid getting down. I think here's the point. Them digging a nest in the gravel. They can pick up stones with that disc. There's lots more minutes of footage of the same. So with that, this is the life cycle of lamprey. So sea lamprey life cycle. We're going to start right here. They build a nest like that in some riffle of a stream, and they lay their eggs then in the gravel. The eggs hatch, and those larvae, those ammocoete larvae, drift downstream into muddy backwaters. Right? And so if you had a question, for instance, on an exam, what's the life cycle of a lamprey? You might want to know this. Ammocoetes are the larval stage. They can live in the mud there, like an amphioxus, filter feeding on detritus or whatever for years. And then they will metamorphose into adults, migrating into a lake or the open ocean, where they might live then as a parasite. So lake meaning one of the Great Lakes, because they're in there now, or the open ocean, where they can attach to another fish and become a parasite. They can live like that for a couple of years, and then they will detach, migrate back to the freshwater rivers and go on that spawning migration, where they then spawn and typically die, but then they then produce the next cohort. So that's the life cycle of sea lamprey. So these are the parasitic sea lamprey, and the larval stage of a lamprey is called an ammocoete. So when we talk about lampreys coming into the Great Lakes, the natural distribution really was the North Atlantic Ocean and inland waters thereof. So for instance, the Admiral Sound in North Carolina. Sea lampreys were introduced inadvertently into the Great Lakes through the Welland Canal that was completed in the 1800s that allowed for the invasion into the Great Lakes. This is actually a canal that bypassed Niagara Falls. So a sea lamprey can't swim up Niagara Falls, right? It's just too tough. So when they built this canal, they were able to get around. So it really connects the Great Lakes to the ocean. The sea lampreys then attacked a wide variety of the Great Lakes fishes, and then they completed their life cycle there. So you have all these lake trout. They have these nice, soft bellies with very small scales that were just perfect for a sea lamprey to attach to. And so unfortunately, they crashed a lot of these fisheries. So lake trout, whitefish fisheries really crashed in part due to these lamprey introductions and following the canal. People try to control these with chemicals, for instance, larvicides to kill the ammocoetes and things like that. They call it under control, but really there's still a lot of lampreys out there. And so here's a video that sort of talks about lamprey control in the Great Lakes. And so this is another video discussing the impacts of the sea lampreys.
Since the 1950s, the Sea Lamprey Control Center has worked under an international agreement with the Great Lakes Fishery Commission and in close cooperation with the U.S. Fish and Wildlife Department towards the delivery of effective sea lamprey control in the Great Lakes.
The fishery in the Great Lakes was devastated through the 40s and 50s, primarily as a result of sea lamprey predation. The governments of Canada, in response to this devastation, formulated the Great Lakes Fishery Commission in 1955. And the primary duty of the commission was to formulate and develop a program to minimize or eradicate sea lampreys from the Great Lakes. Sea lamprey are native to the Atlantic Ocean, and they gained access to the Great Lakes through various ship canals that were constructed in the 1800s. They invaded Lake Ontario in the early 1820s, but were prevented from getting to the other Great Lakes by the presence of Niagara Falls. The Welland Canal changed that, gave them access to Lake Erie in 1921, and from there they spread rapidly to the other Great Lakes. The problem with sea lampreys is that they're a parasite on fish. They subsist on the blood and body fluids of large-bodied fish in the Great Lakes, primarily lake trout and salmon, and they're very destructive. On average, a sea lamprey destroys about 18 kilograms or 40 pounds of fish during its parasitic life stage. People don't normally encounter sea lampreys unless they're out fishing for salmon or other species in the St. Mary's River. Occasionally they'll catch a fish that has either a sea lamprey wound, which are very characteristic, a round circular hole in the side of the fish, or they may even find or catch a fish with a sea lamprey attached to it. The St. Mary's River is one of the most productive rivers in the Great Lakes from a sea lamprey control perspective. We currently estimate that there are about 1 million larvae that make the St. Mary's River home. There are a number of other tributaries around the Sault Ste. Marie area that are very productive from a sea lamprey control perspective and require regular treatment with lampricides. These include the Goulet River, the Batchewana River, and the Thessalon River, for example.
Fisheries and Oceans Canada's Sea Lamprey Control Centre is currently housed inside the Natural Resources Canada's Great Lakes Forest Research Centre in Sault Ste. Marie.
Our program has been highly effective. Since its implementation, we've reduced the populations by 90% Great Lakes wide, and this has led to the rehabilitation of lake trout in Lake Superior, and we're seeing excellent signs of the same thing going on in Lake Huron currently. And we also protect viable recreational fisheries for Pacific salmon, steelhead, coho, chinook, who are also subject to sea lamprey predation.
To learn more about sea lamprey control in the Great Lakes, you can visit glfc.org/sealamp. For Go On Shot TV, I'm Greg Seiler.
And so the sea lampreys, you see this as one of the classic examples of an introduction.
For over 60 years, the Great Lakes.
One of the impacts of exotic species introduction. So something like the whitefish and the trout of the Great Lakes, they had lived there for thousands of years and never had to deal with a sea lamprey, didn't have any defense mechanism against it. And now we see this introduction, and now those fish have to deal with that. And you sort of see then the advantage that an exotic animal has on another that's not prepared. And so this is actually one of the classic examples of impacts of exotic species introductions. And again, they try to use these larvicides to kill the ammoset larvae. So that's the sea lamprey. Any questions there? Excellent. So now we're moving on to the gnathostomes. Now, I would like to show you something here. Look at, let me get my good pointer. There we go. Look at this. I was telling you these other small fishes, like, oh, they're about like 30 centimeters. Sea lampreys like 40 inches or something. This is not 40 inches, right? This is not 30 centimeters. This is the difference between having jaws and not. If you can have jaws, that means you can eat more stuff. If you can eat more stuff, that means you get bigger. And so these things here were substantial. So the earliest gnathostomes. No living descendants. The earliest fishes, they started these placoderms, started around 450 million years ago. No living descendants. Placoderm, plate, skin. These literally are bony plates. These are literally slabs of bone. The whole head is just bone, very thick. Marine and freshwater distribution. We actually believe they might have actually been able to tolerate both saltwater and freshwater, whereas we were talking before, hagfish were only saltwater. Lampreys, you know, they live in the ocean, but they can live in freshwater as well. These earliest ones also might have been able to tolerate both. Diverse body forms and ecology. So once the jaws formed, of course, the first thing you do with those things is eat something else. But it doesn't necessarily need to be meat. You know, you can eat vegetables as well. You know, it's a balanced diet. So there was a lot of diversity there. First vertebrates to colonize the open ocean and fresh water. So a lot of this thing, the idea is it started in the ocean, then went to fresh water, and you'll find out later that then it went back into the ocean. So the fishes had colonized the ocean twice. Length, 6 to 9 meters. That's 10 to 27 feet. So again, these things are relatively big. Their nicknames are the terrible fish. They're the first giants of the vertebrate lineage. They have a persistent notochord similar to the existing coelacanth. Massive head armor and this thoracic armor. So they had armor all along their head and usually like a ring of armor about halfway down their bodies, like this one here. But the tail oftentimes was naked. There was no scales there. They had movable joints in the skull so they could open their jaws extraordinarily wide. And the body was then covered sometimes with some bony scales. Mostly it was naked. They didn't have teeth. These literally are bone. They're bones. They're not teeth. So that's what the teeth are made out of, bone. And so those are the first of the gnathostomes, the placoderms. When this occurred, and the textbook actually discusses this pretty well. They talk about Roemer's hypothesis, the invention or the advent of the jaws, major evolutionary advancement. So look at this. This is like a picture of a side picture of the lancet Amphioxus. Again, here's this wheel organ with these buccal cirri for filter feeding and this branchial gill basket. So food feeding here, gas exchange kind of going on here. When we decided to make jaws up, what we did was we first created a bigger opening, an oral opening for the mouth, kind of like what you'd see in the lamprey, for instance, or the hagfish. And then what we see is the first 2 gill arches are lost. So these 2 white lines here, they're lost. The third in orange then becomes the jaws. And then the fourth in blue here then became what we call the hyomandibula, or the hyomandibular cartilage that supports the jaws. So the early jaws later look like this, and then the later jaws look like this, where literally this comes down and it sort of helps support this lower jaw. In some cases, the third cleft right here behind the orange and the blue, right here, there's a spiracle. And it's a hole in the side of the shark's head that allows it to pull water into its mouth, across its gill, and out through that spiracle. It's an excurrent siphon. It can be an incurrent siphon in skates and rays that live on the bottom. So that way they're not always sucking in sand. And so there's a spiracle right about here between the orange and blue arches in the sharks. Now, if anyone's seen a shark bite, this is very flexible. This jaw can actually pull away from the skull. This white part here is the skull. Here is the eye socket. The jaw itself can actually pull free. It's very mobile. And we'll show a picture of a great white shark biting next week. But they literally can move their jaw out of their head, so to speak, which is pretty crazy. And so this is sort of the idea of how jaws evolved. And it's called Roemer's hypothesis. Again, this major advancement put all the other non-jawed vertebrates out of business. So very important.
Hey, Ben,
So we look
I have a
at,
question for you.
what's up?
Question for you. Do you know of any advances as far as the hypothesis suggesting that sort of those dentine-based scales migrated inward toward the mouth? Has that been pretty much disproven in the last like 7 years?
That I don't know. I don't know. We'll talk about that. Something like that is interesting. I don't know what the determination is on that. But so what Matt's asking is, when we talk next week about elasmobranchs, the denticles, the dermal denticles, the scales of a shark are actually teeth. They literally are teeth. They have the exact same characteristics. So the idea is, if we started off with having teeth on the skin, is that how teeth ended up in the mouth? Or vice versa? I don't know. And so I don't know as far as the status of that hypothesis, who's proven what, but generally speaking, a shark not only has teeth in its mouth, they're also entirely covered with teeth. So the teeth appeared in the mouth and the skin, and I'd say that that's a fossil record issue. Like when we find dermal denticles in the fossil record, we know they were around for a long time, but which came first, chicken or the egg, I do not know, Matt.
Sounds like a good paper topic.
It's I'll expect to see that in this November. That's a good one, Matt. All right, very good. So here we go. Here's some types of jaw suspensions. Jaws allow fish to capture prey. If you can capture prey, you can evolve to consume alternate prey items. So meat and vegetables, not just detritus and muck, right? These things actually, the more stuff you can consume, the more active you can become. You're not a hagfish tumbling around on the bottom. So we have these autostylic jaws. Autostylic is right here. Here's the graphic for it. 1 star. Hyomandibular cartilage is not attached to the jaws. Hyomandibular cartilage, again, was this 3, right? The blue. It's not attached, and you can sort of see, I got a little star here. This is not attached, right? It's freely hanging there. This is stuff you find in lungfishes, dipnoi, the placoderms, other ancestral fishes. Us tetrapods have that too, right? We've just got a mandible that really just slams stuff against the bottom of our skull. It's very primitive. The amphistylic jaw, right here, number 2. This actually has the hyomandibular cartilage, the hyoid arch, connects to the skull. So see these attachments here? Right here. We're now seeing this hyoid arch is attaching to the jaw, right? It's not just hanging loose there. This is in the Crossopterygii. The coelacanth is an ancient fish. It's in the Selachii, so some of the elasmobranchs have it. And then there is the hyostylic, the most advanced jaw support apparatus. The hyomandibular cartilage connects directly and strongly to the skull here and then to the jaw, right? A lot of elasmobranchs have this. The chimeras have this, the rat tails. Actually, these are called holocephali because of this. And then the osteoichthyan fishes. All the other 26,000 whatever fishes have this, right? So a handful have these types. Most of them have this. The most advanced fishes have it. And so really, it's just how many elaborate connections do you have? How well is this jaw attached to the skull, right? And so there's connections here and here. And so these are the jaws. It also allows maneuverability. This jaw here is not very maneuverable, like ours. Your mouth can't come out of your head, so to speak. These jaws can't. They're very loosely attached here with ligaments, and so this whole thing here can push out and down. And I'm going to show you what that looks like here in a second. So any questions there on the types of jaws? Because when you look at the bones, it's one thing to look at the jaw structure itself here. Then we look at the actual bones of the mouth. We can sort of see how this works. So all that jaw stuff I'm talking about is here. This is right here. When you look at the bones of the jaws or the mouths themselves, we have the upper jaw structure in particular varies. So again, like I said, we're vertebrates. Your mandible is literally on a hinge, and all you're doing is chewing. You're just smashing stuff against the bottom of your skull. Think about that next time you eat. That's all you're doing, right? Your teeth are grinding this stuff up, but really your jaw is just pushing stuff up to the roof of your mouth and crushing it against the base of your skull. Very simple. Fish, however, have a little bit more sophisticated way of doing it. So that's sort of what's going on here. This is an example of a trout. rainbow trout. They have what's called a premaxillary, a maxillary, and then a supramaxillary. And they can have teeth on all this stuff. But really all this thing does is this jaw sort of comes down and slaps up, and it sort of chops things. Primitive skull, less derived skull. We look down here at a moon eye. So you're looking at something like cyprinid. We see that these fishes have lost this supramaxillary bone. It's not there. And we see that this premaxillary bone has gotten a little bit smaller, and there might not be as many teeth on it, and that this maxillary has sort of elongated to form a hinge here at the corner of the jaw. When we look then at an advanced fish, a perch or a bass, like a largemouth bass, we find there's this very strong hinge here. This maxillary, here's our mandible, and then this premaxillary is designed so that when this jaw opens, when the bottom mandible comes down, this red maxillary swings forward, and this premaxillary swings forward, and this fish's mouth can open very wide. And when it does so, it can increase the volume of its buccal cavity and create suction, which is suctorial feeding, the pipette feeding. Very powerful feeding apparatus here. This is great for biting onto stuff and chewing it and tearing chunks out like a shark. This is great for just swallowing something whole when it least expects it in a fraction of a second. And then you just bring it down into, we'll talk later, there's a pharyngeal jaw here that pulverizes it. So it just gets it in here, crushes it, swallows it and puts it in the stomach. So here is a video of a sunfish and a largemouth bass feeding with this suctorial apparatus that is so fantastically sophisticated. And you'll see how quick this is. Like it? Yeah. What? Boom. All that. Let's just build that on it. I feel like that's it. Boom. That's being apparatus. If there's another. See how that jaw opens at the mouth and bends forward. Various. So when we say that that's this is what we're talking about. Literally, hey, Sam doesn't even know it. See that when I open the top, see this, the neck goes forward. It really goes to the mouth. Oh, good. I like it. All right, goodbye. Crayfish. So that is this. Oh, let me do this again. Pointer options. That is this again. This hinge here, this maxillary. When that fish opens its mouth, this thing can swing forward, and the premaxillary can come out. Creates a massive vacuum that can pick up a prey item just like that. And when we talk about feeding and digestion, there is a very sophisticated pharyngeal jaw in here that pounds that thing. So even a crayfish, for instance, can get crushed right in here. So fishes actually have two jaws. They've got this oral jaw and this interior one we'll talk about later. So these are some of the different types of skulls. And again, showing you how powerful that jaw as an invention is evolutionarily. So any questions there on jaws? And again, jaw protrusion equals this pipette suctorial feeding. Very big difference here. So, generally speaking, when we look at fish, you can just look at a fish. And based upon that phylogenetic tree I gave you as the first slide for this lecture, you know, we're talking about less derived fishes and talking then about more derived fishes. You can look at it like this. If a fish has a jaw like this, where the tip of the snout extends beyond the edge of the lower jaw, that's usually a less derived fish. So like a sturgeon. This is a lot more ancient than, say, a largemouth bass. So look at this. If the bottom part of the jaw, the mandible, extends beyond the upper part of the jaw, this is an advanced fish. And again, you can see this very prominent maxillary here, knowing that when this fish lowers its jaw, this thing swings forward and the gape of that mouth becomes extraordinarily large. You can also tell that this is a predator, right? This thing here is a predator. So upper is protrusible. Again, this thing swings forward and it can extend forward. So the theme is generally true for most fishes. However, fishes always have exceptions to the rule. This is a sucker, family Catastomidae. Based upon feeding niches, some advanced jaws have evolved based on that strategy. So for instance, even though this is an advanced fish, it has a downturned mouth because these feed on the benthos. It's an adaptation. After this jaw was evolved, suckers evolved to feed off the bottom. And so these things are called suckers as a group because their mouths look like suckers. I mean, it's kind of creative, I guess. But these things have downturned mouths. And so not all, this doesn't apply to everything. You sort of have to understand the phylogeny and the feeding strategies to know where the exceptions are. And we'll talk about those. But generally speaking, this is actually a pretty true trend, right? At least in these two examples. So the suckers are an exception. So with that, I have a story. So this is actually an interesting one. We started off by talking about hagfish. And so we look at the hagfish and we show the video of them producing all this slime. Now look at this. So there's a news article out here that I'll show you here in a minute. But eel skin is hagfish. I mean, does that make sense? I just told you eels are not hagfish. Well, and it really, we call things like eel-like body when really we should be saying hagfish-like body because hagfish came first. But anyhow, the term, people knew what eels were and they like to call things eel stuff and not hagfish stuff. So eel skin wallets, you know, you might have heard this online or something. It's all bunk, right? It's not an electric eel. These things don't demagnetize your credit cards if you make a wallet out of eel skin. And as a matter of fact, eel skin isn't even eel skin. It's actually hagfish skin. So again, we say eel-like body when we should say hagfish-like body, and we say eel skin when we should actually say hagfish skin. But I guess no one would want to buy a hagfish wallet because it just doesn't have the same ring to it. So hagfish skin is very thin and very durable. It's actually more durable than leather made out of like cowhide. And the U.S. Fish and Wildlife Service allows hagfish to be marketed as eel skin because they've been doing it for a long time, like decades, centuries. Hagfish slime also could be the fiber of the future. So I saw the video of these things making all this slime. Not only is their skin better than leather, but as a matter of fact, the slime contains thousands of very thin proteins, 1/100th diameter of a human hair. And these threads are extremely strong when stretched out. And if you dry them out, they're like spider silks. So there's people out there making bulletproof vests out of hagfish skin. There's a lot of like surgical sutures are made out of it. Artificial like mesh for surgical devices are made out of this. Super strong fabrics in biotechnology, things like that, artificial tissues. These are all things people are looking at using hagfish slime for. So despite these things tumbling around on the bottom, blind, in the cold, feasting off of the muck, they're actually kind of interesting and they're useful. And so actually, in some species are actually threatened because of this due to intentional fishing and also unintended bycatch. So there is some concern there for them because they're actually harvested. And so I know people are looking at culturing these things in aquaculture as well so that they can actually acquire the slime. Some people will actually eat these, believe it or not. I've never eaten one, but I will tell you this, when we talk about osmoregulation, the body fluid of a hagfish is the same osmolality as the ocean, which means they're salty. So I don't know how they'd taste. They'd be pretty darn salty. But anyhow, here's a news article that I found from a couple years ago. I think a student pointed this out to me. This is actually pretty, look at this. Thousands of slime eels spill onto Oregon highway in gross gooey crash. This is from 2017, July. So although no humans sustained major injuries in the crash, there were a high number of hagfish casualties. Look at this. These eels are all, or these hagfish are all over the road. This is the pavement. And look at the slime they made. They slimed this car, just like they slimed a shark. So what happened here is police in Oregon were in a slimy situation on Thursday after a truck carrying 7,500 pounds of live hagfish failed to stop at a construction site and spilled contents onto the highway and over surrounding cars. And then they Twittered this. Look at this. These eels are in the ditch on the side of the road. They have a cleanup crew there. That car wrecked. So it says, according to the press, truck driver failed to stop at a construction site along the road, causing one of its containers of hagfish to topple and land in the opposite lane. The container hit a vehicle and caused a 4-car pileup. Then the container's wriggling contents sloshed onto some of the vehicles involved in the crash. And so here, look at this. Again, when we talk about why they call it eel skin wallets, I mean, who's going to go out there and buy a snot snake? Think about that. That's a pretty tough common name to get around marketing. It's like your marketing division. All right, you guys, this is what you got to work with. Snot snake. Go ahead. How can we sell this? So here's a video they posted. Check this out. There's a bulldozer moving the hagfish off the road. And so then they go on to talk then about this. So although they have no jaws or bones, they blah, blah, have 2 rows of keratin-like teeth. And somewhere in here they talk about, he says here, they could capable of filling a 5-gallon bucket with slime seemingly instantly. Is involved in Korean cuisine. Somewhere in here I thought they did talk about, yes, here, hagfish slime is so tough and dense that scientists are looking at using it to create natural lycra. And so that's what they say there. They urged crash victims to scoop the dense slime off their vehicles and throw away their clothes because the hagfish slime is so tough. So there you have it. Hagfish in the news on the road causing a vehicle pileup crash. And so with that, are there any questions? That is the first lecture there on the less derived fishes. I'm going to close this right now. And I'm going to go up here and I'm going to go stop recording. If yes, do you want to stop recording?